My name is Afnan, and we are going to discuss the embryology of eye. So, the eye is formed appears as an pair of shallow grooves on the sides of the forebrain on the 22nd day of the uh, embryal growth. With closure of the neural tube, these grooves form outpocketings of the forebrain, the optic vesicles. This is the optic vesicle here, and this is the otic vesicle for the ear, and this one is for the eye. So, then you can see here in this picture, this is the wall of the forebrain, and these are the optic grooves. These are the optic vesicle. Later the lens play cords form in the mesenchyme, and they invaginate inside, and this is the transverse section of the formation of the optic vesicles and lens play cord. After some time, the optic vesicles begin to invaginate and form the double-walled optic cup. This you can see here is a double-walled optic cup, which has an outer layer and inner layer and a choroid fissure through which the hyaloid artery passes in the intrauterine life and supplies blood to the developing structures of the eye. You can also see here this is the intraretinal space. This is the wall of the brain, and this is somewhat present laterally in the brain, and this is the choroid fissure seen laterally cut. So this is the lens vesicle and hyaloid artery supplying it, right? Now, during the seventh week, the lips of the choroid fissure fuse and the mouth of the optic cup becomes a round opening the future pupil, right? During these events, cells of the surface ectoderm initially in contact with the optic vesicle began to elongate and forms a lens play cord. You can see here we have discussed this is a lens play cord which is invaginating. So during the fifth week, the lens play cord loses contact with the surface ectoderm and lies in the mouth of the optic cup. Development of retina, iris, and ciliary body. The outer layer of the optic cup, which is a pigmented layer, and development of inner neural layer of the optic cup is from more complicated. The posterior four-fifths of the optic cup, because we should be imagining the eye as a circle in which the posterior four-fifths the pars optica retina and the anterior one-fifth the pars ceca retina, right? Posterior four-fifth pars optica retina contains cells bordering the intraretinal space. These differentiate into rods and cones, and rods are more numerous in number than cones. Rods are for black and white vision, and cones are for the color vision. Adjacent to these photoreceptor layers is a mental layer, which is formed by the neurons. This is the outer nuclear layer, inner nuclear layer, ganglion cell layer, outer inner plexiform layers, and nerve fibers from these area form the optic stalk, which then develops into the optic nerve. Here in the figure 20.3, uh, this somewhere retina is here, and these are the layers in the retina. So the optic stalk is like this, develops like this. The anterior one-fifth of the inner layer, the pars theca retina, remains one cell layer thick, later divides into pars iridica retina, which forms the inner layer of the iris, and pars ciliaris retina, which participates in the formation of the ciliary body. Now, the region between the optic cup and the overlying surface epithelium is filled with loose mesenchyme, and the sphincter and dilated pupil muscles are formed from in the tissue. These are formed from the underlying ectoderm of the optic cup. The pars ciliaris retina is easily recognized by its marked folding. It is covered by a layer of mesenchyme that forms the ciliary muscle on the inside and is connected to the lens by suspensory ligaments or zonula. So you can see in this picture right here, This is the lens. This is the neural layer of retina. This is the pigment layer, the outer layer. So here's the ciliary process. These are sphincter pupilla, and this is the more developed ciliary process is forming the ciliary muscles, dilator, and sphincter pupillae. These are all formed from the mesenchyme of the ectoderm. Now we move to the development of lens. After the formation of lens vesicle, cells of the posterior wall began to elongate anteriorly and form long fibers that gradually fill the lumen of the vesicle. By the end of seventh week, primary lens fibers reach the anterior wall of the lens vesicle, and the growth of the lens is not finished at this stage. So here in figure 20.6, we have the this is the lens, and these are the suspensory ligaments through which it's connected to the iris. So formation of the choroid, sclera, and cornea. And of fifth week, the primordium is completely surrounded by loose mesenchyme. This tissue differentiate, and the inner layer forms a highly vascularized pigmented layer known as choroid. The outer layer develops into sclera and is continuous with dura mater and the optic nerve. And the choroid is continuous with and the choroid is continuous with the pia mater, and sclera is continuous with the dura mater around the optic nerve. Now, the eye differentiates in the anterior chamber between the in front of the lens and iris, and behind the cornea, and a posterior chamber, which is anterior chamber is in front of the iris and the cornea, and from the iris to lens is the posterior chamber, and this is filled by aqueous humor. So cornea is formed by epithelial layer derived from the surface ectoderm, substantia propria or stroma, which is continued with the sclera, and epithelial layer which borders the anterior chamber. The iridopupillary membrane in front of the lens disappears completely. The posterior chamber is the space between the iris and the anteriorly and the lens oscillatory process ST. Posteriorly, we have discussed this. From the anterior chamber, the fluid passes through the scleral venous sinus. So, aqueous humor is produced in the posterior chamber. It passes through the scleral venous sinus canal of Schlemm in the anterior chamber to the aqueous veins, and then to the vein is drained by the venous drainage, and the blockage of this movement of fluid causes glaucoma. Glaucoma can be open angle or closed angle. The development of vitreous body. Mesenchyme not only surrounds the eye primordium from the outside but also invades the inside of the optic cup. It forms the hyaloid vessels which supply the blood in intrauterine life, and the interstitial spaces of the network is filled with a gelatinous substance called the vitreous body, which are after that obliterated. Hyaloid vessels are obliterated and disappear during fetal life, leaving behind the hyaloid canal. Hyaloid canal is a reflection seen in the anatomical gross anatomy of the eye, which passes through around central to the transverse section. Now, optic nerve. Optic cup is connected to the brain by the optic stalk, which has a groove, the choroid fissure on the ventral surface. These groove has hyaloid vessels. When the hyaloid vessels are obliterated, not necessarily obliterated, but during the seventh week, the choroid fissure closes and narrow tunnels form in the optic stalk, form inside of the optic stalk. This optic stalk, narrow tunnels forms, and the nerve fibers grow, and the optic stalk becomes the optic nerve. Now, this is the structure of the optic stalk. This is the optic stalk. This is the outer layer, the lumen, nerve fibers, hyaloid artery, and choroid fissure. When it's completely formed, the optic nerve, it forms the optic nerve, and the optic nerve has inside the central artery of retina and a vein and the optic nerve fibers. These are optic nerve fibers, this is the artery and vein. So, moving to the pathologies, coloboma may occur if the choroid fissure fails to close. Choroid fissure fails to close. Uh, this occurs in the seventh week of development, and if it's really in the iris only, it is called the coloboma iridis, and it can occur with because it's formed due to the mutation in the gene. So it can also cause renal problems. Iron pupil membrane may persist instead of being absorbed during formation of anterior chamber. So congenital cataracts. Lens become opaque in the intrauterine life. Can be seen in the mothers which have been suffering through measles, mumps, and rubella, or any other viral diseases during pregnancy. But thanks to the development of MMR vaccine, this has been eradicated, and the hyaloid artery may persist and form a cord or a cyst. The microphthalmia is eye is, the eye is small, and anophthalmia is the absence of the eye. Congenital aphakia is the absence of the lens, and iridia is the absence of the iris. These are also due to the mutation of the genes. Cyclopia, single eye, and synophthalmia, fusion of the eyes. This is synophthalmia. This is aniridia. Aniridia is basically the absence of the iris. So cyclopia is due to the loss of the midline tissue. So it is associated. It can be associated with a brain defect called holoprosencephaly. Cerebral hemispheres are partially or completely merged because the midline tissue is not present, and the lateral sides merge together, and this thing appears. It can follow up to the brain, or it just remains anteriorly. So this is for the embryology. Thank you so much for listening.